Hello, everyone. I hope everyone is doing good. Today, we're going to talk about DNA technology. DNA technology is a technology that has to do with the DNA manipulation, DNA changes, and every biological activities or procedures which the DNA would go through. The focus question here is what is genetic engineering and why is it useful? Genetic engineering, genome restriction enzyme, gel electrophoresis, recombinant DNA, plasmid, DNA ligase, transformation, cloning, polymerase chain reaction, transgenic organism. All of these are the new vocabulary which we going to talk about in this lesson. DNA is a genetic material of all organisms. Every organism has DNA. It is what stores the genetic information of all the organism, and it's composed of two complementary chains of nucleotides. Complementary means when you have A, we have complementary bases. We know A, Chargaff's rule says A goes for T and C goes for G. So a DNA would have two complementary chains like this. So one chain is going to face the other one and it's going to go one around each other. So if you got A here, you have a T here. If you have a C here, you have a G here. And the way it continues. So this is what is being meant when you say two complementary chains of nucleotides wound in a double helix. So in a double helix means you're going to have it in this way. It's going to be wounded like this. So to the end of it. By about 1970, researchers had discovered the structure of the DNA and were able to determine the central dogma that information is being flown from a DNA to RNA to proteins and not vice versa. So you will now have RNA to DNA to proteins. That is not possible. That is against the central dogma. So the central dogma says the movement is from a DNA to RNA and to proteins. However, scientists did not know much about the functions of individual genes. So it took the scientists time before they could know what the function of each individual gene was. So the situation changed when the scientists began to use genetic engineering or technology to manipulate the gene of an organism. So when this gene is manipulated, then they would be able to knock out a gene and then try to see what the function is. For example, if a gene is knocked out and suddenly the effect is being felt, like you don't feel the effect of that gene anymore, then you will know this is the function of the gene. Let me give an example. We know the hair usually grow maybe after some time. So when you bob it, so let's say, for example, you have an organism, and what did you do? You decided to knock out a specific gene and you cut the hair. So then after you knock out the gene, after cutting the hair, you notice the hair did not grow back. It didn't grow back, or it took a longer time before it grew back. So automatically, that would tell you that this gene, which I just knocked out, is the one responsible for the formation of the what of hair growth. So it is the one that actually triggers the growth of the hair in the body. So this and many other means or ways is how the scientists kept on studying the genes one after the other. So it involves manipulating the DNA of one organism. So we manipulate the DNA of one organism so that we can insert exogenous DNA, that is to say the DNA from an outside organism. So we can embed, or we can put into it the DNA of all the organisms into the DNA of the organism. Genetic engineering can be used to increase or decrease the expression of specific genes. So expression of genes is something that actually control almost all the activities in the human body. When, for example, when there is a need for a specific gene is going to be expressed, and the expression means that there would be transcription, and also after that there would be translation. So that's what we call gene being expressed. So it has many applications in from the human health to agriculture. An organism's genome is a total number of DNA present in the nucleus of each cell. So when you say the word genome means the total number of DNA which an organism has. In order to study the specific gene, DNA tools can be used to manipulate DNA and also to isolate genes from the rest of the genome. So in order to study a specific gene, if you want to study a gene, so the DNA tools, which we're going to talk about later on, can be used to manipulate. Manipulation means to change, to fix something which is not somewhere in that place. And it can also be used to isolate genes from the rest of the genome. Now we're going to move on to restriction enzyme. What is a restriction enzyme? If you check your dictionary, the word restriction, to restrict means to minimize or to not to allow someone to get to a place. But in biology, restriction enzyme is actually been used to cut or cleave the DNA at specific sequences. So when we use restriction enzyme, we use it to cut, or we use it to cleave DNA sequence at specific locations. Restriction enzymes are used as powerful tools for isolating specific genes or regions of the genome. So we can use these genes to isolate specific genes or regions of the genome. Now, among the DNA tools which we want to talk about is the restriction enzyme. And today we want to move on to the examples of the restriction enzymes we have. A good example of the restriction enzymes we have is EcoRI. EcoRI or R1 got the name because it's found in E. coli. So it's found in E. coli, and this organism, it has the specific type of the E. coli is the R13, and that's how we got R, and a 1 means the first one we got. So EcoR1 is a restriction enzyme that specifically cuts the DNA containing the sequence GAATTC. So whenever this restriction enzyme is going around in the DNA, anytime it comes across this GAATTC, it cuts in between the G and the A. So it cuts from here. So leaving these as sticky end, and also you're gonna cut from the other side. So you're gonna have the same GAATTC. So it's gonna cut also from here. So all of this would be restriction ends, would be sticky ends. So the sticky ends are single-stranded DNA sequences at the end of the fragment. So they can be joined together with complementary sticky ends too. So any complementary base that has the same sticky end with a sticky end for EcoR1 can be easily joined to it. Another DNA tool or DNA methodology or technology is gel electrophoresis. Gel electrophoresis is using electric current to separate DNA fragments according to the size of the fragment. When electric current is applied, the DNA fragments move towards the positive end of the gel. So what would usually happen is the unique pattern which has been created is based on the size of the what of the DNA fragment, and is going to be compared to the known DNA fragments for identification. So this is the way in which the gel electrophoresis works. It's one of the most important technology used in the science lab today. DNA fragment from different sources can be combined to make a new DNA. So we can have DNA fragment. Let's say this is one DNA fragment. Yeah, stick in here. This is another DNA fragment. 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 So, and let's say has stick in here. Say, so you can decide to use a ligase as an enzyme to join this one to this one to this one to this one to this one. So all of these fragments can be joined together to produce one specific DNA molecule. So when this happens, because these are all cut from different DNA, from the different DNA molecules, so when you combine them together, you call that a recombinant. As the name implies, re means it was not combined before. It was combined before, it was whole before, it was being restricted, being cut, and now it's being combined again. That's what the word recombinant means. So recombinant DNA is placed into the bacterial cell for study via the vector. So we're going to use a vector to carry this the recombinant DNA, and we're going to enter it into the bacterial cell so that we can study the vector properly. Common vectors include viruses. Sometimes we use viruses to send these new or recombinant DNA to an organism. So viruses can be used. The virus would actually inject the vector into inject the new recombinant DNA into an organism. We can also use a plasmid. A plasmid is a small circular double-stranded DNA molecule that occur naturally in bacteria and is also found in yeast. DNA ligase is a cellular enzyme that helps to attach the recombinant DNA to the plasmid. This is a good example of what we're talking about. This is a plasmid DNA vector. So the plasmid DNA vector has been restricted in this area. So now this is a genomic DNA and is also been restricted in this area. Mind you, if it's going to be restricted, it's going to be cut by the same restriction enzyme. It's going to cut them at the same position, so they would have the same sticky end. So what would happen is now you're going to join them together here. So now you have the recombinant DNA, which is having the DNA plasmid, the vector, and the genomic DNA. So now it's going to form one single organism, which is got from a combining two different genetic material. Now let's talk about gene cloning. To make a large number or large quantity of this recombinant DNA, which we just produce, what do? The bacterial cell are mixed with the recombinant plasmid DNA. So because the bacterial cell easily take up the recombinant DNA through the process of transformation. Transformation is a procedure which the bacteria goes through by sucking in or taking in the recombinant DNA into its own DNA. So when the transformation happens, the is we have a bacteria that has its own DNA, plus the recombinant DNA. So we can expect to see different behavior or different features in the organism. Bacteria that take up the plasmid make copies of the recombinant DNA. So the bacteria that take up the plasmid, they make copies of the recombinant DNA during the cell replication. Large number of identical bacteria containing recombinant DNA can be produced through the process of cloning. If you produce identical bacteria that has a recombinant DNA, because if you introduce the recombinant DNA into the bacterial cell, not all of them would transform. Some of them would, and some of them would not. So let's take a look here, everybody. So this is a plasmid. This one you see here is a recombinant plasmid DNA. This recombinant plasmid DNA has what we call ampicillin, what do you call it, DNA in it. So it has an ampicillin DNA in it. So what is ampicillin? Ampicillin is an antibacterial medicine. So now we're going to inject it into the bacterial environment. Some of the bacteria undergo transformation. They absorb the recombinant DNA into their own cells, while some other ones they would not take it in. So what happens is there is a replication of the recombinant of the bacteria that took the recombinant DNA and they copied the recombinant DNA plasmid. So we have them to reproduce in large number. So the cells that refuse to take the recombinant DNA, the cells that refuse to take the recombinant DNA. So the bacteria that got transformed, they multiply in large number. And also the ones that didn't get transformed, they also multiplied in large number. As you can see, this ones here that does not have the plasmid inside them, they did not get transformed. This one also did not get transformed. And the ones with this plasmid, they get transformed. So now what happens is you're going to have a plate. You're going to have a plate that has ampicillin in it. So these plates would actually be loaded with ampicillin antibiotics. So you want to try to see if there is some organisms that can actually cope with the ampicillin in it. So what did you do? So you're going to put the bacteria in with the ampicillin. So when you did that, you found out that the organisms, the bacteria, which has a recombinant plasmid DNA with ampicillin in it, they survived. And the ones without the ampicillin, they died. So that gives you the understanding that these bacteria already had the ampicillin gene in the body. So this is how we do it. So now the next thing is we want to talk about DNA sequencing. What is DNA sequence? Scientists study the DNA sequence study the sequence in the DNA fragment. So what do they use? They use DNA polymerase, fluorescently labeled. So they also use fluorescently labeled nucleotide bases, and what they use electrophoresis. So how do they do these? What they do is this: they know they want to sequence. They want to have a large number of the gene. So they want to sequence the whole genome. So how do they do this? So they're going to give each nucleotide a specific coloration. So when these colors appear after the sequencing, they would be able to read through the coloration to produce the genetic code, the C, the T, the A, and all the rest. So this method is not very sophisticated. We use a sequencing machine to do it. And all of the necessary things, once you put it in a sequencing machine, allow the process to proceed, and you're going to get your result. Now, polymerase chain reaction is called PCR. Is once the sequence of the DNA fragment is completely done, so a technique called the PCR is being used to make millions of copies of the specific region of a DNA fragment. So that specific region you got here, you can see it's very short, right? You're going to want to amplify it. So what you want to do is this is going to produce a normal DNA for you with a double helix, right? So then you would want to increase the number. So what happens is the method which you're going to use to increase the number is called polymerase chain reaction, called the PCR. It can be used to make millions of copies of specific regions of a DNA fragment. PCR can copy or amplify a single DNA molecule numerous times for use in analysis. This we already talked about in the classroom. We had a virtual lab, which we showed you all of this. PCR is performed by placing the DNA fragment to be copied, the DNA polymerase, 4 nucleotides, 2 short single-stranded pieces of DNA, which is called the primers. You put all of this in the test tube. So the PCR is a biological version of a copy machine. During each PCR cycle, the reaction mixture is heated. So when you heat the mixture, it helps to separate the double-stranded DNA. And when you cool it down, it allows the newly formed DNA strands to combine with the old ones. So this helps to increase the number of the fragments. So the DNA polymerase then would add nucleotides to form new molecules. This is the example what I'm talking about. So the recombinant DNA technology, we have the first one is a denaturation. So in the denaturation step, what happens is DNA strands separate by heating. So you're going to heat it up to above 90 degrees. So you will see the hydrogen bond between the nucleotide bases is going to break up. So once they break up, then you're going to find a heat-resistant polymerase. So this heat-resistant polymerase would be used to make new nucleotide bases. Mind you, before that, you're going to put the primers in the edge of each of the section. So this is the top and this is the bottom. So this is where the restriction enzyme actually cut it. So you're going to put the primers here. So then the nucleotide bases in the process will start adding up, and that's called extension. So annealing is when the primers combine with a single-stranded DNA. So extension is when the new nucleotide bases are coming up and they attach into the complementary bases. So the end result is going to be what? It's going to be 2 new DNA which is identical to the one we started with. I think that is clear. So biotechnology is the use of genetic engineering to find solutions to problems. Organisms with genes from other organisms are called transgenic. When you say an organism is transgenic, the meaning is it's got a gene from an organism in its own body. So transgenic animals, plants, and bacteria, they've been used a lot today for research in medicine, in agriculture. Transgenic animals can be used by the scientists to produce most transgenic, to study diseases, to improve food supply, to improve health, and also the potential sources for organ transplant. Today, I was reading an article where people are growing a mouse they're growing mice in the lab. These mice have organs of humans in them. So they try to produce organs for transplant because a lot of people are on queue for organ transplant. We don't have what can suffice everyone. So we're trying to find a way to produce organs in the laboratory using some transgenic animals. Transgenic plants are frequently genetically engineered for resistance against insect and viral pests. Sometimes ago, people usually use pesticide to kill these pests that destroy the crops. But today, we can manipulate. You can use transgenic procedure to inject the gene, which is making the plant to be resistant to the insects and viral pests. All the transgenic plants are designed to reduce allergic reaction in humans. They contain increased vitamins and minerals, and also they resist extreme weather condition. For example, if you live in places like Sweden, where in winter it's frozen. So if you have plants which you want to grow crops, but unfortunately everywhere is frozen, you can have a gene that would allow the plant to not to really freeze on time, and this plant will be fine even when all the plants are frozen in the environment. Transgenic bacteria can be used to produce insulin and growth hormones. They can help to slow the formation of eyes on crops too. They can help to clean oil spills, where, for example, where there's oil spillage on the ocean, we can generate some bacteria and take them to those places to help to clean up by breaking down the hydrocarbon composition of the oil and allow it to dissolve in the water. We can also use it to decompose garbage. So we can put some transgenic organism in the garbage so to help break down the contents of the garbage. So this marks the end of the content. So, but just you can go through these questions yourself. Which of the following tools or processes is not involved in genetic engineering? So restriction enzyme is very involved in it. Here, human genome, gene cloning, DNA sequences. Of course, you're going to think that B might be the correct answer. So you can answer the rest yourself. I think it's pretty easy. So I wish you all the best in your finals, and we're going to see you again.